Hey guys, welcome back for oxidative phosphorylation. That includes the ETC and chemiosmosis. This is where we're going to make by far most of our ATP. So far, we've only made 4 ATP. If you remember, we made 2 in glycolysis and we made 2 in the Krebs cycle / citric acid cycle. So most of the energy from the food that you have consumed has been transferred into 2 molecules, FADH2 and NADH. So those are, remember, both electron carriers. So both of them are going to drop off their electrons. They're going to donate their electrons to the ETC, the electron transport chain, and that's going to power ATP synthase. And it's going to do that via oxidative phosphorylation, which I know is a big fancy term, but just think of it as this: using oxygen to phosphorylate ATP. All right, so how does that all work? What you're seeing here, here's NADH. Dropping off its electron. Here's FADH2 dropping off its electron. So here is our electron transport chain. So if I follow this electron, where does it end up? Well, it ends up here at oxygen. So oxygen is pulling electrons towards it. Now, why does oxygen do that? Well, oxygen, if you remember, is highly electronegative. So that's why it hogged electrons in its bond with water, right? So oxygen being highly electronegative is going to pull electrons toward it through the ETC and eventually going to create water. So one of the things that goes into the ETC is oxygen, and that is going to turn into water. So water is a product. The other 2 things that go in would be NADH. There'd be 8 of them, and then there would be 2 FADH2s. And their again job is to drop off their electrons. So what are those electrons doing in the meantime? Well, their electrons' job, what they do is we're using the energy that's in those electrons. And what you're seeing here is we're pumping hydrogen ions or protons into the intermembrane space. And what that does is creates a huge high concentration. In other words, it's creating a concentration gradient. And that has a lot of energy in it. So you can see all these hydrogen ions up in here. Those want to diffuse down their concentration gradient. So we have a huge high concentration, right? High to low. And they do that through an enzyme called ATP synthase. So we're going to use that protein, ATP synthase, that enzyme, and that enzyme is going to create a ton of ATP. So where did the ATP really come from? Remember what this is called. It's oxidative phosphorylation. We're using oxygen. That's going to help us create this huge gradient, which is called chemiosmosis, and that's going to move down through ATP synthase and make ATP. So that's kind of the process, how this works. Now, if you remember, we talked a little bit about last unit about why the mitochondria has all kinds of folds on its inner membrane. And the reason why is because on that inner membrane, look, here's our inner membrane, that's where the ETC is. So if we have a lot of surface area, that just means that this can happen all over in more areas. Okay, so it creates more surface area for this to happen, which allows us to create a lot of ATP. So at the end of the chain, electrons, again, are passed to oxygen. It is good to point out that NADH has about a third more energy than FADH2. We create about 2 ATPs per FADH2 molecule. We create about 3 ATPs per NADH molecule, so it does hold a little bit more. But those electrons would move through the ETC and eventually go into oxygen, which creates water. So this is the actual chemiosmosis bit. So again, here we have our high concentration of hydrogen ions. They're going to move down their concentration gradient through ATP synthase, and that's going to create ATP. That is chemiosmosis. So that chemiosmosis piece, again, fancy science term, right? All it is we have this high concentration. So we have a concentration gradient. They move down their concentration gradient through ATP synthase. So if you remember what osmosis was the diffusion of water. Diffusion of water is high concentration to low, so this is just chemiosmosis, so it's chemical osmosis. So rather than moving in water, we're using chemicals to do this process. So that's kind of why it's called that. So at certain steps along the electron transport chain, there will be that pumping of hydrogen ions from the matrix into that intermembrane space. This is creating that high concentration. So the resulting gradient, that has a lot of energy in it, and that's what drives chemiosmosis. We're going to use the energy from the electron to make this gradient, and then we're going to move that through ATP synthase to make ATP. That whole process, the creation of the gradient and then the moving through ATP synthase, is chemiosmosis. Okay, so we're going to use that gradient to drive chemical work. So this is that overall process again. Here's NADH, here's FADH2. They're dropping off their electrons. Their electrons move through the ETC, eventually going to oxygen, which creates water. Okay, but while those electrons are moving through, you can see it here, here, and here. What are they doing? They're pumping hydrogen ions into this intermembrane space. So let me grab my pen here a second. We have a whole bunch of these inside of here. In other words, we have a high concentration, a low concentration. So these things want to move down their concentration gradient through ATP synthase, and that's going to make a ton of ATP. So two parts to oxidative phosphorylation: the electron transport chain, chemiosmosis. Both of them together drive oxidative phosphorylation, using oxygen to phosphorylate ATP, creating a ton of energy for your cells. So during respiration, most energy flows in this sequence. We flow from glucose to NADH or FADH2, which bring their electrons to the electron transport chain, which uses their energy to create that proton motive force, which is basically the high concentration of hydrogen ions, protons, and that is used to make ATP. So overall process here, we have glycolysis, breaking that down into pyruvate, which is made into acetyl-CoA in the link reaction, enters the citric acid cycle. Then we use the NADH from the glycolysis and the citric acid cycle to drive the ETC. The ETC is going to make a lot of ATP through oxidative phosphorylation. Now, about 40% of the energy in a glucose molecule is transferred to ATP during cellular respiration. That makes approximately 38. Now, on average, it's usually between 34 and 38, which is kind of complicated to explain. But again, it's usually between 34 and 38 ATP in order to get that to work. So let me just bring this up a second. Oh, shoot. Guess not. Doesn't work anymore. Anyway, we will draw the process instead. I'll see if I can find a video, and I'll post that to Canvas if I find one. Oh, where am I? Hold on. And go back to this slide to draw the process. All right, so here's the whole process of cellular respiration. Let me grab the green here. So in glycolysis, we break down glucose into pyruvate. Okay, plus we make 2 ATP, 2 NADH. Okay, that pyruvate, so this would be pyruvate. There's 2 of them. This would be glucose. Pyruvate is going to enter through the link reaction. And that's going to create, let me grab a different color. It's going to make 2-acetyl-CoA. Those reactions together in the Krebs cycle are going to make 6 CO2s plus 2 FADH2, 2 ATP, and 8 NADH. Now, okay, that's where we were at the last time we drew this. Let me grab a different color. Now we're going to get into oxidative phosphorylation, which is ETC plus chemiosmosis. Okay, so what do we use? If you remember what goes into this process, we use NADH and FADH2. So those two are going to go in. They're going to drop off their electrons. So how many of those did we start with? We started with 10 NADH and 2 FADH2. Okay, so we have some proteins that make up the ETC here. Those two are going to drop their electrons off. Okay, and their electrons are going to move through this process. They're eventually going to end up at oxygen, which is going to make water. Okay, then what we have is this protein called ATP synthase. Okay, now remember, as these electrons that are going through the ETC, they're going to, wrong arrow. They're going to pump hydrogen ions and create this huge concentration. So we're going to have a high concentration in here. Now, the only way that they can move out is through ATP synthase, and that's going to create ATP. Okay, so two pieces to this. The ETC is this part right here. That's where the electrons move through these proteins, to oxygen and create water. The chemiosmosis piece is the creation of this gradient, and then the eventual movement through this enzyme ATP synthase to create ATP. So if I look at the overall process, we have the ETC down here. We have what goes in, oxygen, and 10 NADH, 2 FADH2, and then what are we going to make? We make water plus about 34 ATP. Okay, so you can see why we said makes about 38 ATP. 34 plus 2 plus 2 make your 38 ATP. So that's the whole process. Again, we break down glucose. We do this through a series of steps, but eventually, what is the goal? Our goal is to break down glucose and to make a whole bunch